Hello everyone, I'm Jona from the University of Toronto, and today I'll be taking you through the BioSequence Ontology from molecular structure, an ontology for first-order reasoning across gene sequences. To construct an ontology, one must first ask, what is a gene? It is commonly understood as a sequence of nucleotides in DNA or RNA that code for a gene product, say a protein. The fact that it is a sequence highlights two things. That is, it is comprised of discrete elements, and that order matters. If we look to the diagram, figure 1 shows an arbitrary gene sequence. We can proceed to take an arbitrary subsequence, say ATGC, as shown in figure 2. ATGC can be further broken down into either ATG or TGC, and further into AT, TG, or CG, and so on. This points towards a mirror topology where combinations of elements can form subsequences, and combinations of subsequences into even bigger sequences, as illustrated in figure 4. However, it is noted that this is not a classical Mereology, as not every pair combination is possible. For instance, TC is not a possible combination, as G is in between T and C. This property is known as convexity. With this intuition, it leads to our three primary research questions, namely, how can we represent gene sequences as molecular structures? It is understood that a gene has a 3D molecular structure, but it is also understood and interpreted as a sequence. Moreover, what is the representation for linear and circular gene sequences? Beginnings and endings are unclear in a circle, and direction less clear. And finally, what is the mirror topology of gene sequences? Of course, building an ontology for gene sequences is not a novel endeavor. However, we feel that our ontology will fill a gap in understanding yet to be addressed by the gene ontology or the sequence ontology. That is, to simultaneously represent a gene as a sequence and a molecular structure, providing a complete and rigorous definition of parthood, with explicit axioms for concepts of between-ness and directionality, using the aforementioned definitions of chemical structure and parthood to perform primitive first-order reasoning about gene sequence function, and finally, using this derived understanding of gene function to reason about gene processes, such as transcription and translation. These needs are further expressed in concrete competency questions. Examples testing the intersection of concepts are shown in the green boxes between the clouds. For instance, the start codon is directly upstream of the 5' UTR. To answer the question, how is the 5' UTR and start codon related, understanding of parthood and chemical structure of the sequence is required. To test competency regarding parthood and function, we can ask the question, what do the DNA codons ATT and TTA code for? This is because to be able to differentiate between ATT and TTA, the ontology needs to have a concrete notion of directionality. Further, to test understanding regarding function and process, one can ask what is the intron or exon in the given sequence. The intron is removed via splicing during translation, whereas the exon will encode a part of the final mature RNA. And hence, the ontology needs to really understand the function and process of translation to answer that question. And finally, an example of a question that tests competency under intersection of gene chemical structure and process would be what are conservative replacements for the conserved sequence in the gene coding for the H1 histone protein. However, we know what our mereology should do, but how should we define it? These three axioms capture the essence. For instance, the first axiom states that two elements are connected if and only if they have a mereological sum. This guarantees that for any two nucleotides that are adjacent in a molecule, there exists a gene sequence that contains their sum. The second axiom states that topological sums exist whenever mereological sums exist. And the third and final axiom guarantees that any gene sequence is ultimately decomposable into nucleotides, which functions the axioms of the mereology. With that understanding, we can construct axioms for two different types of DNA and connected substructures, namely linear versus cyclic. One may wonder why so much emphasis is placed on a seemingly simple concept. However, explicit axioms for linear and circular betweenness translates to an accurate and consistent understanding of where 5' begins and 3' ends, as well as the general composition and overlap between sequences. Now that we have our ontological commitments, we can use them to guide ontology design. To fulfill our need for representation as both a sequence and a molecule, the BioSequence Ontology will serve as an extension of the molecular structure ontology, inheriting its signature and concepts such as mole and skeleton. As the focus of the ontology is gene semantics, the lowest level of abstraction of concern would be the functional group. To go into further details, such as atoms or bond types, could impediment understanding and workflow efficiency. Yet by inheriting most, this minute level of information would still be available and essentially part of the ontology under the hood. Perhaps rather conveniently, most represents its molecules as molecular graphs, which can be modeled as path graphs. The bijection between path graphs and convex ordering was proved in another previous paper, a Mereology for connected substructures. We were able to take advantage of this result and incorporate it into the BioSequence Ontology. As shown, this is the betweenness axiom for cyclic betweenness, the first one, and the latter a betweenness axiom for linear ordering. Finally, we consolidate this information and utilize it to axiomatize functional subsequences to form the signature. For instance, the definition of an open reading frame is defined here as a reading frame that transcribes to an RNA or translates to a protein. Definitions of reading frames and proteins are further defined in the BioSequence Ontology, but have not been included in the set of slides. However, we can guarantee that all definitions are directly or indirectly defined in terms of sequence, skeleton, and/or functional group. Finally, to complete the circle, the same competency questions guiding the design of the ontology will be used to evaluate the performance of the ontology. All of the competency questions here have been rewritten in first-order logic and terms within the signature of our ontology. For instance, for question 1, how is the 5' UTR and start codon related? We have essentially changed it into, for all start codons, does there exist a 5' UTR whereby they are connected by the mole relation? For the second question, essentially we have rephrased the question to ask if ATT and TTA code for the same protein. If the ontology is able to differentiate between ATT and TTA, it will understand that they will code for different proteins, namely leucine and isoleucine. However, questions 3 and 4 are incomplete. This is because the intron and exon are defined based on the result of processes that change the sequence, such as transcription, translation, and splicing. As such, in the future, we aim to create mappings to existing gene sequence ontologies to increase our knowledge base and lower barrier to usage for other researchers. Moreover, we are working on building the biosequence process ontology based on the process specification language, PSL, to enable representing change across gene sequences and provide representation for transcription, translation, and splicing. In conclusion, we have successfully built a reusable gene ontology extended from the molecular structure ontology. We have defined separate and explicit axioms for cyclic and linear betweenness, and finally axiomatized a non-classical Mereology that was the basis for other gene part definitions. Thank you, and we will now open the floor for questions.